In our previous video, we have learned about the different types of transposons. The transposons or the jumping genes, they are the stretches of polynucleotide sequences that change their position within the genome. And the basis of their classification is the mode of transposition. That is how they change their position within the genome and the intermediates that are involved in the process of transposition. So in this video, we will try to understand the basic methods of transposition of the DNA segment. Now, before we come to our topic, let us quickly revise the major types of transposons. Transposons, they are indicated in the genome by the presence of flanking regions. They are the inverted repeats, or they may be the long terminal repeats, the LTRs, and there's a certain category of transposons in which the LTRs, they are replaced by the UTRs, that is the untranscribed uh, stretches of nucleotide sequence, and such type of UTRs, that is the untranscribed terminal repeats containing transposomes, they have got a poly A tail at the 3' region. Now, whether these transposomes, they transpose by means of directly the DNA, that is the class 2 transposomes or the DNA transposomes, or the class 1 transposomes, which are the retrotransposomes, which involves the reverse transcription. Transposomes of both these classes, that is the class 1 retrotransposomes and the class 2 DNA transposomes, they may be autonomous, that is they have the gene segments for the enzymes, either the transposases or the replicases, or they may be non-autonomous and depend upon the enzymes provided by some autonomous element. Now, the transposition mechanisms for the major uh, type of transposomes, they are shown over here. They involve transfer of DNA segments directly, as in case of DNA transposomes, or may take place via the RNA intermediates, as is the case in the class 1 transposomes, that is the retrotransposomes. Now, in DNA transposomes, it may be by the simple process of cut and paste, or maybe copy and paste, as is seen in electrons, where there is a strand transfer and then it is replicated in both the segments. However, in case of retrotransposomes, every time it is the copy and paste type. Now, the cut and paste mechanism or the conservative transposition, it involves two enzymatic activities, the endonuclease activity and the ligase activity, that is cutting the DNA strand from in between by breaking the phosphodiester bond and forming the phosphodiester bonds at the target site. And the enzyme that is responsible for both these activities, it is the enzyme transposase. It excises the transposome segment from the donor DNA and then it generates nick at the target site and integrates the DNA segment at that particular point. Now, in case of the replicative transposition, the single strand from the donor, it is cut, and this single stranded donor, it integrates with the uh, recipient DNA molecule or at the target site, and then after, these stretches of single DNA strand which are present in the donor site as well as in the recipient site, both of them, they form the replica, that is DNA replication takes place. Both these strands present in both the DNAs, they act as the template, and we get a copy of this transposome at both the sites, the in the donor DNA molecule as well as in the recipient or the target DNA molecule. Now, during conservative transposition, that is by the cut and paste transposition, it may be simply that the segment of DNA, that is the transposome, it cuts from the donor DNA segment and it integrates at the recipient DNA or at the target site. Now, in such a case, at the donor site, the flanking repeats, the flanking direct repeats, they join with each other, they ligate with each other, and a footprint is left at this point. A footprint which denotes the site where the DNA transposome was present. However, in certain cases, the donor DNA, it gives its transposome to the recipient DNA, but it itself generates a similar DNA segment or the same transposome by making use of the homologous chromosome. That is, the cut ends, they do not ligate directly. Instead, the repair of the excision site, it takes place by the homology dependent gap repair, and for the purpose, the homologous chromosome that is present in the cell, it is made use of. So in this manner, although the method was that of cut and paste transposition, but the donor site as well as the recipient site, both have got a copy of the same transposome. For transposition of retrotransposomes, first of all, transcription takes place of this transposome, and as a result of transcription, RNA is produced. This RNA, it has got gene sequence for the reverse transcriptase enzyme. So this enzyme is translated, that is by means of translation, this enzyme reverse transcriptase is produced, and by the activity of this enzyme produced, reverse transcription of this RNA, it takes place, resulting in the formation of a DNA strand, and then after, the second strand of DNA is synthesized, keeping this first strand as the template. Now we have got the double stranded DNA molecule of this transposome part, the retrotransposome part. This DNA, it inserts at the new site or the target site, and thus transposition takes place. So we say that in case of retrotransposomes, it is a copy paste mechanism via the formation of RNA, and it involves transcription as well as reverse transcription steps. So, in case of non-LTR retrotransposomes, the RNA, which is transcribed at the donor sequence, it reaches the target site where a nick is generated just near this poly T region. Now, the poly A sequence base pairs of this RNA, it base pairs with the poly T sequences, and it serves as the template for the formation of your uh, DNA molecule by means of reverse transcription. And for this purpose, this T region, it acts, or it provides the free hydroxyl group. It acts as the primer. And in this manner, once the DNA stretch is formed, this RNA, it is replaced by the second strand, the complementary strand of DNA, and ultimately what we obtain is a DNA transposome at the new site, and on both the ends they are present these direct repeats, which are a characteristic feature of transposomes of both the class 1 as well as the class 2 transposome. So these are the various methods by which transposition of DNA it takes place.